Now come to consequences of endosmosis. I think you remember in my previous class I have discussed what is endosmosis. The process of osmosis which causes inflow or inward movement of a solvent molecule into a cell or tissue from an outer medium is known as endosmosis. Clear? Suppose this is a container in which we take water or hypotonic solution. And this is a membranous egg. Or a plant cell. Clear? So, in the outer medium, I have taken a hypotonic solution or pure water. And here, you know that cell is a very small microscopic structure. Alright. So, I have also written it is a membranous egg. What is membranous egg? Now, you know that egg of bird is externally covered by means of a hard shell of calcium, calcareous shell. Alright, this is basically calcium carbonate. So, if you put a shelled egg in a dilute solution of hydrochloric acid or sulfuric acid, clear, the calcium carbonate portion of the eggshell will react with the acid solution and will dissolve. Clear? And you know, if you boil an egg and then try to remove the shell, immediately beneath the shell you find a membrane. Is not it? So, that is the egg membrane. So, if we remove or dissolve the eggshell by putting it in dilute solution of acid, the eggshell dissolve and the egg is left with the membranous covering only. And remember that egg membrane is semi-permeable in nature. Clear? Also, in case of plant cell, you know that plant cell is externally surrounded by a definite cell wall, and inside the cell wall, clear, there is a protoplasm. Clear? So, a plant cell is divided into two parts. The non-living part of the plant cell, that is the cell wall, and the living part of the plant cell, that is the protoplasm. Clear? And the protoplasm is surrounded by means of a membranous structure. The outer part of the protoplasm become membranous in nature, that is commonly known as a plasma membrane. This is cell wall. And in my previous class I told you that cell wall is permeable in nature, but the plasma membrane is semi-permeable in nature. One thing more which is very important here, that if you observe a healthy plant cell under microscope, high power microscope, clear, you can easily see the cell wall and you can easily see the protoplasm surrounded by the plasma membrane. Okay? And in a healthy plant cell, there is always a narrow gap between the cell wall and the plasma membrane. Clear? Under high power microscope, a definite gap between the cell wall and the plasma membrane is seen. Now, come back to this experiment. This is a hypotonic solution or water, and in this hypotonic solution we have placed a membranous egg or a plant cell. What do you expect? There will be movement, inward movement of a water molecule. Alright, there will be inward movement of water molecule from the outer medium inside the membranous egg or inside the cell. If it is a plant cell, clear, if it is a plant cell, now drop this membranous egg come to the plant cell. If it is a plant cell, when the process of endosmosis takes place, there will be continuous addition of water. Clear? There will be continuous addition of water in the protoplasm of the plant cell. Is not it? The water from outer medium is continuously entering into the plant cell, passing through cell wall and the plasma membrane. That will definitely increase the water content of the protoplasm, and that in turn will cause the expanding of the protoplasm. Clear? Due to increase in the volume, the quantity of the protoplasm will increase, and this increasing volume of the protoplasm will exert pressure onto the plasma membrane. Clear? The plasma membrane is living and elastic in nature, so it will be stretched. Clear? Due to the pressure from inside, the plasma membrane will be stretched. Clear? And slowly the plasma membrane will stretch to such an extent that it will get adpressed, or it will fix to the inner part of the cell wall. Clear? So that means the gap between the cell wall and the plasma membrane will disappear. Clear? So in the process of endosmosis, when there will be continuous inflow of water from an outer medium inside a plant cell, clear, there will be increase in the volume of the protoplasm, and this increasing volume of the protoplasm will exert pressure, clear, in outward direction, that is to the plasma membrane, and then plasma membrane being elastic in nature will stretch and ultimately will come in contact of the cell wall, will get adpressed with the cell wall. And under this situation, if you observe the cell under microscope, you will find no gap between the cell wall and the plasma membrane. Clear? This condition of the cell is known as a turgid condition. Clear? This condition of the cell is known as turgid condition, and the phenomenon is called turgidity. All right. So turgidity defines the state of a cell after the process of endosmosis, which exhibit increased volume of the protoplasm with stretched plasma membrane, rendering no gap between cell wall and the plasma membrane. Clear? So turgidity defines the state or condition of a cell when after the process of endosmosis, clear, due to increased volume of protoplasm and the stretched condition of the plasma membrane, clear, no gap is left between cell wall and the plasma membrane. Clear? Now, turgor pressure. So due to turgidity, why the plasma membrane is stretched? Because plasma membrane is experiencing some pressure. The plasma membrane is experiencing some pressure from within the cell, that is from the protoplasm of the cell, as a result of which, as a result of this force or the pressure, the plasma membrane gets stretched. And when the plasma membrane after stretching come in contact of the cell wall, then the protoplasm along with the plasma membrane, clear? The protoplasm along with the plasma membrane will apply force, clear, onto the cell wall. And this force which is exerted from within the cell, within a turgid cell, clear? This force which is exerted from within a turgid cell and experienced on the surface of the cell wall is known as the turgor thrust. And when this turgor thrust or force is measured on unit area of the cell wall, it is known as turgor pressure. So remember, turgor pressure is an internal pressure developed in the turgid cell, clear, after the process of endosmosis. Clear? So this is the consequence of endosmosis. So what is endosmosis? I am briefing once again. Endosmosis is the process of osmosis which allows the inflow on inward movement of the solvent from an outer medium into a cell, tissue or a system. Clear? Due to this endosmosis, there will be, if we are considering a plant cell, then due to this endosmosis, there will be continuous increase in the volume or quantity of the protoplasm of the living cell, and this increasing volume of the protoplasm of the living cell will exert a pressure onto the plasma membrane surrounding the protoplasm, and plasma membrane being living and elastic in nature will stretch, clear, will expand outward and ultimately will come in contact of the cell wall, as a result of which finally no gap will be left between cell wall and the plasma membrane. At this stage, the cell is known as a turgid cell, or the phenomenon is known as turgidity. Clear? And then as a result of turgidity, a turgid cell apply an outward pressure onto the cell wall. This pressure exerted by a turgid cell, clear, onto the cell wall is known as turgor pressure. Clear? Now come to consequences of exosmosis.